Hello, my name is Leho Tedersoo, and I'm working in the Mycology and Microbiology Center in the University of Tartu. The mission of our lab is to understand the microbial biodiversity globally, and also what are the key drivers of this diversity. We are also interested how biodiversity is related to ecosystem functioning and ecosystem services. Our lab itself is mostly a molecular lab that is capable of doing various biodiversity-related molecular analysis with a focus on long-read sequencing, which gives particularly accurate and high-resolution results for biodiversity.
That's very interesting. What have been your key achievements to date so far?
Well, I think that the main achievements are related to global biogeographical analysis of soil microbes, including both bacteria and fungi. We have built the first global maps of fungi and bacteria, and very recently we also estimated the relative endemicity patterns of fungi in certain areas of the world, and we also evaluated conservation needs for fungi globally.
That's very exciting. Tell us more about your research laboratory, how big it is, what are the values in your lab, what is important to you as a group leader?
Well, our group is consisting of 5 PhD level researchers and also 10 PhD students who are all linked to this broad field of microbial and also soil animal biodiversity, ecosystem functioning. We are a very international team. Our group has personnel from 8 nationalities, and we are also valuing good relationships within the team and international linkages both within the group and between different teams. So we are quite social groups. We have our own internal, let's say, free time meetings, etc.
I see. 10 PhD students, this is pretty huge research laboratory. If I remember by our scale, 9 students that we had, that was a big lab. How do you decide when you give for the projects? How are those students, how they are supervised, how do they lead the projects? What is essentially the culture in the lab?
Well, each student is assigned to 2 supervisors. One of them is like a more broad scientific mentor, and the other one is more specialized on these particular types of analysis, so that the student can ask very specific questions. Of course, the main duty of the student himself or herself is to solve those, let's say, cutting-edge topics by itself, but the supervisors are to provide great help and advice when the student has to pick a way.
I see. So if we are talking about the current state of the lab when we are in the COVID era, so it's been already since quite a while, and now we'll have the new global event, which is the war in the Ukraine. Is there any effect of these two events on the research in your lab? Did it change somehow?
Yes, well, it's a very good question, and actually both events have affected us quite strongly. First of all, COVID generated a lot of uncertainty, not only like financially, but also socially, because links between people started to degrade when there were short lockdowns, although in our university it was not as severe as in many other European countries. So we paid a lot of extra attention to this social coherence, and I tried to be still at work. So whenever students came, we could discuss their problems. And to be honest, this war in Ukraine, this has affected us more strongly because we have several students actually from Kharkiv region in Ukraine, and we have tried our best to support them and also to support Ukrainian collaborating researchers. So, for example, right now we are looking for opportunities to provide extra funding to our Ukrainian collaborators who are either still in Ukraine or in other parts of Europe.
I see. Yes, this is very interesting, and this is exciting to hear how the Estonian people support the Ukrainian people. My personal background, I also coming from Ukraine, but I spent in Germany already 10 years, so I left the country immediately 1 year before the first revolution essentially started. Right, yes, so we stand with Ukraine, that resonates a lot. Let's continue to the next question. We've spoken so far about the research in your laboratory. We've spoken about the culture and the way how the students work, as well as the external factors that influence. For you as a group leader, the leading research institution, what do you want to achieve for yourself next? Are there any obstacles that are still staying in the way to those achievements?
Well, first we have to see that Estonia is a very small country, and the University of Tartu itself is quite a small institution because of the total population of Estonia. So we certainly have some financial issues, especially regarding those, let's say, ultra high size projects, because there is certainly some kind of a ceiling into which we can't break through. But in general, I would say that financing of research is reasonably good in our country. So personally, I do not have worries for myself, but I worry about younger colleagues because we have some issues funding, let's say, this consolidator stage young researchers. But my personal ambitions would still preparing some kind of global overall analysis of microorganisms from very different kinds of substrates and understand how all these systems function and which factors actually are related to promoting this biodiversity. But also a very important mission for myself is to promote this research field more broadly in my country and also globally. and also to provide sufficient boost to our former students and postdocs so that they can get good, well-paid positions either in academia or in industry, either in Estonia or worldwide.
Yes, thank you so much for this. Would there be anything you would like to share with our viewers, anything that they should know about, maybe even those who have never heard about the discipline, and there is a lot of value in it. Is there anything you would like to share with them?
Well, if they are getting somehow involved, they will realize that it is a very fascinating and intriguing topic. And nowadays there are also a lot of connections to industry, especially biotechnology and pharmacy. So picking such a PhD or master's topic might be favorable for future finding a very nice job.
Good. Thank you so much for this. We appreciate this interview. Thank you for the information shared.